This is achieved by treating the bacterial cells of plant or animal tissue with enzymes like lysozyme bacteria, cellulase, centrifugation with proteins such as histones. RNA can be removed by treatment with ribonuclease. Proteins can be removed by protease. Ultimately, purified DNA is precipitated out after the addition of chilled ethanol is cut into number of fragments by inspecting the length of DNA. Then it will bind to the DNA and cut each of the two strands of the double helix at specific points. These are called molecular scissors. Over 250 restriction enzymes have been isolated so far. When linear DNA is treated with restriction enzyme, a large number of DNA fragments are formed. The resultant fragments are separated by gel electrophoresis. Finally, the desired DNA fragments are selected by Southern blotting technique. Once the desired fragments of DNA genes are obtained, they are inserted into a suitable vector to produce indefinite number of copies of genes. This is known as gene cloning. A cloning vector acts as a vehicle to carry the desired gene. Several types of vectors are used in recombinant DNA technology like plasmids, phages, etc. The enzyme DNA polymerase helps in replication. If the process of replication of DNA is repeated many times, the DNA segment can be amplified to approximately billion times. That is, 1 billion copies are made. Such repeated replication is achieved by use of thermostable DNA polymerase, which remains active during high temperature induced denaturation of double-stranded DNA. The bacterial cell is treated with EDTA, ethylene diamine tetraacetic acid, along with lysozyme enzyme to digest the cell wall. Then the bacterial cell is subjected to centrifugation in sodium lauryl sulfate solution to separate the plasmid. The isolated plasmid DNA is cut with same restriction endonuclease enzyme. The enzymatic cleavage cuts circular plasmid into a linear molecule having sticky ends. The two sticky ends of this linear plasmid are joined to the ends of the desired gene. The enzyme DNA ligase joins the complementary ends of plasmid DNA with that of desired gene by covalent bonding to regenerate circular hybrid called recombinant rDNA or chimeric DNA. The recombinant plasmid is transferred into a suitable bacterial host cell, generally E. coli, by a method known as transformation for the expression of desired gene. The cells into which the recombinant DNA are inserted are called transformed cells. Example, if rDNA bearing gene for resistance to an antibiotic, example ampicillin, is transferred into E. coli cells, the host cells become transformed into ampicillin-resistant cells. Only transformants will grow on agar plates containing ampicillin. The transformed recipient cells will die. The ampicillin resistance gene is called selectable marker. Bacterial cell walls are not ordinarily permeable to such recombinant vectors, but reaping in dilute solution of calcium chloride renders the bacterial cell wall permeable to the recombinant vectors. Inside the host cell, this rDNA starts replicating. The transformed cell will begin to grow on medium and divide as separate units. The replicate vectors in each cell are passed on to daughter cells, giving rise to clones.